Okay, we're going to explore biotechnology. This is the last part of our genetics unit. So if you're talking about biotechnology, the first thing to do is to be able to extract DNA and then be able to manipulate DNA. So the first thing that we have here is a website that you can go to do a virtual DNA extraction. It's University of Utah. It is an interactive website. You can go to and it'll work you through the process of DNA extraction. And then when you get to the end of DNA extraction, you're going to end up with something that looks like this. Now, normally, if we were in the classroom setting, we would do DNA extraction from banana. I do have that available to you. I will post it online and you can do it at home. You just need a few things. Isopropyl alcohol, which unfortunately isopropyl alcohol is hard to come by these days. You need salt, dishwashing liquid, or shampoo. I usually use shampoo. Um, Baggies, stirrers, bananas. You don't need a whole lot of materials, but again, I'll share that with you on Classroom. But basically, in order to be able to manipulate DNA, to work with DNA, you have to first get DNA out of a cell. And again, this website will work you through that. Now, once you have the DNA out, you may want to make cuts. And to cut pieces of DNA, you use enzymes. They're called restriction enzymes. So it's a special group of proteins that will cut DNA at specific sequences, so specific nucleotide sequences. So if you look at this little chart of some restriction enzymes and what the source here, we're looking at some bacteria like E. coli that can be cut. And if you look at the places where they cut, this one cuts anywhere you see the sequence of GAATTC. So there we see it again, GAATTC. Remember, 5 is top, 3 is bottom. And it cuts between the G and the A. So this Echo R1, which is kind of common, you hear Echo R1 used a lot, it makes something called a sticky end. And I'll show you what a sticky end looks like in just a bit. But if you look down here, some of them make what are called blunt ends. And the big difference between a sticky and a blunt end is whether or not there's a little bit of hangover. See, there's a little bit of a hangover there, or whether or not the cuts are straight down. So the blunt ends, the cuts are made without any overhang. So there's nothing to take these two little segments of DNA and join them. Can't join them with anything. A sticky end can be joined with another piece of DNA. So because of the universal nature of DNA, A with T and C with G, if you cut a piece of DNA and there's this little bit of overhang, then you're going to be able to join it with another piece of DNA. Now, gel electrophoresis, and I'll have a little video clip kind of showing you how gel electrophoresis works, but it is something that can be done with DNA segments once they've been cut with restriction enzymes. And DNA itself has a negative charge, so you can place the DNA in these little wells. So this is called agarose gel. So it's kind of like Jello, but more kind of like solid Jello. Um, and you could place the DNA in here, zap it with some electricity, and this takes about an hour or so. And then the DNA fragments are going to move towards the positive end. Now, the larger pieces are not going to move as far because this is kind of like a stiff jello that this has to move through. The DNA physically has to move through it to the other side. The smaller pieces will go farther. And then you end up seeing this pattern that I would say you probably seen somewhere on a television show, or maybe even seen a picture of it at some point. You've seen one of these before. Now, what this resulting is a restriction map. So this is a result of gel electrophoresis. And this restriction map can be used for lots of different things. This one we could see is probably dealing with different types of bacteria, and we could see the different restriction enzymes that were used. Sometimes in some wells they were used alone, and others, a couple of them were combined. Um, so every individual is going to make a unique restriction map, unless you have an identical twin. Okay, so let's say we've got some fragments of DNA that we have separated and we want to test them, and we know that a lot of this is happening right now where you've got certain segments of DNA that you want to work with and see what you can do with it, but maybe you don't want to use your only sample. So you want a lot of samples. And we're going to look at a process called PCR that will allow you to make a gazillion copies of DNA. So PCR stands for polymerase chain reaction, and it produces millions of copies of specific sequences of DNA. And again, you can see I've got a little bit hyperlink here. So everything that I have on here is a hyperlink. I will either attach the link to the website. In this case, it's a link to a YouTube video that you can watch that shows you how this PCR process works. So PCR, the polymerase chain reaction, uses something called Taq polymerase to help build new DNA strands. And this is just isolated pieces of DNA, isolated genes that you want to test, that you need to test. Okay, DNA fingerprinting. What is DNA fingerprinting? So DNA fingerprinting is basically a type of restriction map. So it's the result from gel electrophoresis. And with the DNA fingerprinting, I mentioned before that every person has a unique DNA fingerprint. And everyone has a specific DNA fingerprint because everybody is made through sexual reproduction. So you've got two parents with their own genome, so all of their genes. So we know for every chromosome that you inherit, you have these two alleles for that chromosome. So it makes a great amount of potential DNA that we could test. And you don't have to look at the whole entire genome of a person. You can just look at small sections. But everybody has a unique genome or unique DNA fingerprint unless you're an identical twin, because identical twins come from the same fertilized egg. Now, what a DNA fingerprint can be used for, forensics. So one of my favorite things, crime shows, mysteries, who's the suspect, who did it. And we could see in this little picture here that we have a sample from a crime scene. So collected DNA could come from anything, can come from hair, can come from saliva, can come from shed skin cells. Kitty. And then we have suspect A and suspect B. And then we look at their DNA fingerprints. And if you look at the crime scene evidence, who has the DNA fingerprint closest to that? Well, suspect B. Guilty. Can also be used in paternity testing. And this paternity testing, this is commonly what you would see on the SOL when it comes to DNA fingerprints, is having the parents and then figuring out who are the children of those parents. And the way that you would figure it out is to see who has most most in common. So you're just looking at these little bars, these little DNA fragments, to see how many of those fragments match up with the parent. And usually in those cases, you don't have mom and dad that you're looking for. Um, you've got the parents' information, or you might have just moms, or you might just have dads, and you're trying to figure out which one is their kid. And usually it's not with people either on the SOL. It's usually cats, dogs, horses. Okay, this crazy mess is looking at base pairing sequences that are common in different organisms. So this is kind of a really broad picture here that might eventually turn the um, Linnaeus's classification system of how organisms are classified, how new species are determined, kind of turn that on of its head, because this is looking at the commonality between the base pairings of different organisms. So this is comparing zebrafish, mice, dogs, humans, and frogs. All right. So genetic engineering is changing an organism's DNA. It isn't grafting, it isn't transplanting. It's actually changing the organism's DNA. So it changes at the level of the chromosome. So a lot of different things that can be done with that. One is cloning. And cloning is the production of a genetically identical copy of a cell or of an organism. I've got a couple of little things here for you to look at. One of them is Click and Clone, and it's going back to the University of Utah website, and it's a little interactive tutorial on cloning, and then just a little video clip on how cloning can work. Now, cloning itself, if the price is right, if you've got enough money, you can clone your favorite horse or your favorite dog or your favorite kitty. And then you have to look at the nature versus nurture argument. So, yes, this organism has the same exact DNA, but will it have the same exact temperament? Will it have the same exact personality? Probably will not. But that's kind of like the big argument when it comes to cloning, even though we can and it's not very successful, should we? Recombinant DNA is interesting. Now, recombinant chromosomes, you might hear that term when you're talking about meiosis and crossing over. When we're talking about recombinant DNA and genetic engineering, we're talking about putting together genes from more than one organism. So, again, sticky ends. And because of the universal nature of DNA, I can put together two different genes from two different organisms because A goes with T and C goes with G, always in DNA. Now, plasmid is a single circular piece of DNA in bacteria, and it's used oftentimes as a vector or a vehicle of transmission of genes during genetic engineering. Transgenic, this is a term for organisms that have genes from other organisms. I've got a video here. I'm sure you're going to notice it's going to be about a cow, but how cows are transgenic, how they've received genes from another organism by way of recombinant DNA. These are all examples of some transgenic organisms, and I have a little website that you can go to and look at some of these. So going beyond a glowing pig that's transgenic for jellyfish DNA, or a glowing tobacco plant that's transgenic with lightning bug DNA. And then I have here a little video clip on the Human Genome Project, which is a project began in the 90s, a universal uh, project to map out all of the chromosomes. So taking all 23 different types of chromosomes in humans and figuring out all of the genes. Where are all the genes? What are all the genes? Now the process is figuring out what do they do? How can they interact? So I've got a little video clip, so just work your way through this and play around with the tutorials and see what you think.